So thank you very much for the invitation to give this talk here. I kept to my original title, even though I'm not gonna diverge from disease tolerance, but I'm gonna try to tell you a story on disease tolerance. And all the work I'm going to show you was done at the Gulbenkian Institute, which is in the outskirts of Lisbon. So, to start very broadly, we're interested on events that can disrupt homeostasis, and evolutionarily, most probably, the majority of these are associated with infection. But we also know that tissue injury or stress and malfunction, independently of infection, can also signal this type of events. So we have very good sensor mechanisms that tell our body that homeostasis might become disrupted, and we react to that by an inflammatory or immune response. In most cases, immunity is required when it's infection. In other cases, inflammation might be sufficient. And the whole system actually has evolved to return the system to homeostasis. So when this works, we don't even notice it because it's operational and there's no major signs of sickness. However, there can be there are very significant trade-offs of this response in the sense that it can actually lead to metabolic dysfunction, tissue damage, or even to disease and death. And a clear case where this occurs is when the trigger is actually a blood-borne infection, for example, that triggers both an inflammatory and immune responses, leading, for example, to sepsis. And now we'll give some examples to that. Now, we all know that we have very good systems of defense against infection, and we very use, in particular immunologists, to relate to resistance to infection. So resistance to infection is one of these mechanisms that tries to restore homeostasis in response to infection, and it does so with a very simple strategy, which is to detect the pathogen, and eventually neutralize it, destroy it, or if it's not possible to destroy it, to expel it. And by eliminating the source of the problem, this is a very good way actually to restore the system. Now, there's overwhelming evidence that this is a major defense mechanism, if nothing else. The use of vaccines and antimicrobial agents such as antibiotics have just been one of the major human revolutions. So there's no doubt of the benefits of this defense mechanism. However, this is not able to explain everything. And perhaps what's missing is this notion that there is a parallel, but at the same time interconnecting defense strategy that aims exactly at the same thing, which is to restore homeostasis, but it does so via a completely different strategy. So it doesn't target the pathogens. Actually, it does not carry a negative impact on pathogens. And instead of what it does, it's to limit the amount of tissue damage or other fitness costs that can be caused to the host. And we refer to that as disease tolerance. And the reason to bring this word of disease tolerance is actually to pay respect to the people that first used it, because this is a notion that came from host microbe interactions in the plant literature over a century and a half ago, and they refer to it as disease tolerance. Obviously, in the context of immunology, tolerance is a very charged word, but we felt that actually it was important to pay tribute to the first people that discovered this. So there's many ways actually of representing this. This is an imaginary plot, but actually we can make the real plots out of it. So if you would plot health or fitness, this is actually a very healthy individual, and this is actually an individual dying. And we're plotting it against pathogen burden. And the person that came up with this type of way of seeing evolution of infection was actually David Schneider, and the original reference for it is actually here. So as a parasite grows in its host, one can imagine that until a certain pathogen load, there's actually no fitness costs to the host. If it keeps growing, actually, the health of the host will decrease. And what's strange with these curves, and they're counterintuitive, is that they reverse to the left. We usually see graphs going from the left side to the right side only. And the reason for this is that because you have resistance to infection, reducing pathogen burden, and so over time, as pathogen burden decreases, so does health decrease. And there's variations on this, as you will see. And this is the phase that we call the pathogenic phase of the disease. In this arbitrary graph, this individual is actually almost dying. And if you want to cure infectious diseases, actually what you should aim at, it's at establishing this recovery phase that brings back the individual to his initial health status. And you can imagine the tremendous amount of variation on this. And I'm just going to go through some of them. So everything that brings this curve from the right side to the left side we refer to as resistance. I put on purpose a resistance very low here, but it's coming from the right side to the left because I want to be a little bit provocative and tell you that you can diminish actually pathogen load without having any impact on health. And disease tolerance actually is anything that brings actually the graph from the lower part into the upper part. And that's actually what saves individuals from infectious diseases. So one can imagine actually genetic variations for resistance to infection and something like this. This is an individual that is very resistant. Actually, his immune system is very good at clearing the pathogen, but he actually does get sick, but it recovers. This will be an individual actually that is less resistant. He is not immunocompromised, but the pathogen load grows actually to higher levels. And perhaps as a cost of that, actually, it gets more sick, it almost dies here. And this is an immunocompromised individual that lost actually resistance to infection and actually dies from the infection. So these will be cases of genetic variations for resistance to infection. For disease tolerance, it's slightly different. So this is an individual that is actually very tolerant to an infection. So the pathogen load can be very, very high, but yet he doesn't get sick. If you imagine this is the entry to the hospital, he is walking on the street. This is an individual actually that carries the same pathogen burden, maximum here 10 to the 9, but he does get very sick. And this is an individual that actually has no or very little tolerance to the infection. He carries exactly the same pathogen burden, but actually he dies from it. So what are the bases for this disease tolerance? So we think disease tolerance relies on stress and damage responses that sustain the functional output of parenchymal tissues, being those parenchymal tissues critical to maintain homeostasis. So these systems are not actually self-contained into parenchymal tissues, but they're absolutely critical for these tissues to keep functioning and maintain actually homeostasis in the infected host. The idea is that these tissue damage control mechanisms, initially what they aim at is to provide metabolic adaptation into these tissues to actually compensate for the different forms of stress and damage that are occurring, either as a direct result of the presence of a pathogen or indirectly because of immune-driven resistance mechanisms. If this doesn't work, but I'm not going to talk about it today, actually damage does occur, and there's a second layer of mechanisms that also contribute to tissue damage control, which are important actually to enforce disease tolerance. So a schematic view of these would be that you have different stress responses. These are actually they are sensors for different forms of stress, for example, for redox, oxygen pressure, osmolarity, and so on. These circles here are genetic programs that get actually activated in response to these different forms of stress. And as you will see, actually, they're actually overlapping between themselves, and this is actually important for some notions, for example, that have to do with the fact that creating a certain level of tolerance to one type of stress provides you actually protection against other types of stress. In any case, the outcome of these stress responses is actually to provide metabolic adaptation. And the general notion here is that you need to adapt to that stress by maintaining the core functions of cells or tissues in detriment of everything that is accessory. So this is not a normal functioning of a cell or a tissue. This is an emergency situation, but the cell must adapt to that. And the molecular basis for this type of mechanisms actually are fairly well understood in other contexts, and we try to put them in these reviews here. So we think this is a critical component of tissue damage control that ultimately provides disease tolerance to a variety of infections. So in the context of this talk, I'm going to focus in a very particular component of this type of responses, which have to do with iron and immune metabolism, and how the regulation in the host of iron and immune metabolism impacts on the in the establishment of disease tolerance to infection. So IM is this molecule that is here. Usually it doesn't rotate like that. The only reason why I put it rotating is that you don't forget about it. And the important thing about it is that it contains an iron atom in the middle. So this is a very evolutionary conserved way of maintaining iron inside a protoporphyrin ring, which actually controls to some extent its reactivity. And why is this important? Well, it's important because about 80% of all the iron that is available in mammals actually is inside IM groups. And this is kind of staggering because people, when they talk about iron, they're always thinking of this form, which only accounts for 20-25%. It's never free, actually. It's always bound to something. And for the IM is exactly the same thing. So where can we find this IM? Primarily in hemoglobin. The second biggest pool is myoglobin. And then a variety of other IM proteins, such as cytochromes, account for the remaining. And this is very compartmentalized. So hemoglobin in red blood cells, myoglobin in muscle cells, and this for other cell types. So just in a snapshot, how do we control iron metabolism systemically in mammals? Key to these functions is actually hemophagocytic macrophages. They're always looking for any sort of stress or damage that appears on the surface of red blood cells, and they take up 10 to the 6 of these damaged red blood cells per second. So this is actually a staggering mechanism. So the whole aim of this is to recover the iron, which is inside IM. So they have a transporter called HRG1 that pushes the IM from the phagolysosome into the cytosol. Then IM oxygenase 1, which is an important molecule for disease tolerance, actually takes out the iron, either stores into ferritin, but the idea here is to bring it through ferroportin, which is the only iron exporter that we know, so that it can put the iron back with transferrin and transferrin receptor, and guess where it goes? It goes back into the IM biosynthetic pathway. So the whole idea is to actually take the iron from IM and put it back where? Primarily on red blood cell progenitors, but eventually in muscle cells and other cells. This biosynthetic pathway exists in every single cell. We not actually C. elegans and, for example, some parasites, such as trypanosomes, are IM auxotrophs, but actually we are not, mammals are not, so every single of our cells can produce this. So why is this important? I'm going to bring back the red blood cells because they're the biggest reservoir of iron, and there's and actually if they get damaged and if they're not captured by hemophagocytic macrophages, they can release hemoglobin, which is a tetramer that contains 4 IM groups. They dimerize spontaneously once outside the red blood cell. And there's a variety of events actually that can trigger this. So for example, mutations such as the sickle mutation, vasoconstriction, clotting, activation of both adaptive and innate immunity, changes in pH and osmolarity, or actually the action of pathogens, because microbes and pathogens, they really require iron to proliferate, and this is a great source of iron, so they've evolved a variety of systems to go and capture this iron. So why should we care about this? We should care about it because of the staggering number of red blood cells that we have in the circulation and the very high content of hemoglobin. So a very low level of red blood cell lysis that would pass completely unseen clinically actually generates a tremendous amount of biologically active tetramers and eventually dimers of hemoglobin. So once you have dimers of hemoglobin, you get auto-oxidation of hemoglobin. But in addition, if you have an innate immune response where you have ROS, actually these dimers actually release IM. And this generates labile IM, which you will see actually is tremendously important for disease tolerance. So to go back to this slide, what I'm going to tell you today is that this is probably one of the mechanisms, but by no means the only one, by which actually inflammation and immunity can result in this type of damaging events. While tissue damage control mechanisms, today I'll show you those that rely on metabolic adaptation, must be operational to return the system to homeostasis. So the first molecule I'm going to tell you about is IM oxygenase 1, which is actually an IM catabolizing enzyme. It's very evolutionary conserved. It takes out IM. It uses 2 electron donors to do that. It releases carbon monoxide, which is a remarkable gasotransmitter that has very potent anti-inflammatory and cytoprotective properties, but I will not talk about them today, and actually generates iron, which is actually toxic. You will see that this is a conundrum that will be solved by the expression of ferritin, and it generates a third molecule called biliverdin, which is very important, but we will not touch. So I mentioned this very briefly. We're interested in sepsis and these events in the context of sepsis. These are the clinical definitions of sepsis. And we're particularly interested on this one, which is at the intersection of these different events. So it's an infection with these clinical outcomes, but in addition, a component of multi-organ dysfunction syndrome for which we don't understand the pathogenesis. I will not talk about septic shock in particular today. So a few years back, we've shown that actually this labile IM that gets generated from cell-free hemoglobin is actually critical for the pathogenesis of sepsis, which allows us actually to propose that the expression of hemoxygenase 1, which we've also shown in that paper, actually is a critical factor to determine whether you have high disease tolerance or low disease tolerance. Now, I told you very briefly as well that by catabolizing IM, and the important component of IM is really the iron inside, you're just releasing the iron out of this protoporphyrin cage with generation of byproducts, which are important by themselves. But this is actually very, very reactive and can trigger, for example, by the phantom chemistry, if it reacts with H2O2, it triggers these forms of reactive oxygen species that can trigger oxidative damage. Now, to overcome that, this reaction must be coupled with the upregulation of a molecule or of a protein complex called ferritin, which is a very interesting a set of proteins. Is both a heavy and a light chain, and the aim here is actually to sequester the iron and transform from 2+ into 3+. We call this iron nucleation, and it does not allow it to react and generate free radicals. So it doesn't clear the iron at all, it just stores it and brings it back if needed be. So the question I'm going to try to address with you is, does regulation of host iron metabolism by ferritin impacts on the establishment of disease tolerance to sepsis? So the systems we used are fairly straightforward. First of all, we use normal CFSM like 6 mice, and we expose them to this fairly brutal type of approach, which is cecal ligation and puncture. So we take the cecum, we do a measured ligation, and we just puncture, and they do a very, very acute peritonitis because they get exposed to their own microbiota. So this is a very severe polymicrobial infection, and we do it on purpose because we don't want this to be pathogen specific. So we want to have a fairly strong number of pathogens there. So the first question we ask is whether ferritin is upregulated in this situation. We see it by protein that there is a very significant upregulation of protein in response to cecal ligation and puncture as analyzed in the liver. If you go and look in more detail in the liver, ferritin is in green here. This is a normal liver. If anyone can answer this, it would be very interested, because some hepatocytes, this one express very high levels and this one doesn't express at all. It's a little bit expressed on Kupffer cells, but there's a very significant variation on the ferritin expression throughout the liver. But once you expose to CLP, that you have this very significant upregulation, primarily on hepatocytes, not to say that macrophages are not expressing it. So the second question we ask is, what would happen if you don't have ferritin expression in both components, in macrophages and in hepatocytes? So we use these MX mice, which are inducible deleter primarily in hepatocytes and in leukocytes. We expose them to cecal ligation and puncture, and first of all, we check that actually the deletion is operational in the liver, so there's no cells that express ferritin there. And if we look for survival, we see now that actually in order to survive this very strong polymicrobial infection and survive the development of sepsis, you must express ferritin, because if you do not express, actually, you get a tremendous mortality. I should notice actually that we're treating these animals with antibiotics, and we can discuss that afterwards, and that's very important for the type of conclusions that we're taking. Now, if we look at pathogen load in these mice, and this is actually a fairly difficult thing to do, we look at aerobes and anaerobes, and we went very thoroughly in the peritoneal cavity, which is the initial focus of the infection. We went to blood, liver, and spleen, and we see absolutely no differences in the amount of colony-forming units that are there. So this is by definition disease tolerance. What I mean to say is that ferritin is actually providing disease tolerance or conferring disease tolerance to sepsis. So we can go back to this type of slides and say that, like HO1, ferritin promotes disease tolerance, while decrease of ferritin compromises disease tolerance to sepsis. So what is the molecular mechanism for this? So we went for the obvious type of things. We said, okay, ferritin might be acting as an immunoregulatory molecule, and by doing that stops this cytokine storm, which is known actually to contribute to the pathogenesis of sepsis. And we see absolutely no difference for a fairly big number of cytokines, which actually can be used as a fingerprint of the overall innate immune response that is operational here. So we don't think actually ferritin is acting as an immunoregulatory molecule. So afterwards we said, well, this has to be tissue damage because ferritin is known to be actually very cytoprotective, and in particular in the context of TNF-mediated programmed cell death. So we did serology to reveal actually liver injury, muscle injury, genital injury, or kidney injury, and we see absolutely no difference between these two situations. So I would say that desperation, if it doesn't kill you, it helps you. And that's exactly what happens here. And then Sebastian Weiss, that was doing this project, decided to measure blood glucose levels in these animals. And what he found, to our astonishment, was that ferritin, which I will remind you is a key molecule to regulate iron metabolism, actually carries an impact on glucose metabolism. So we saw that actually, this is the mice that express ferritin, and they drop on glucose as measured 24 hours after the initial infection, and then they tend to recover. This is the total number of mice. This is the mice that are alive, and you can see that when the mice are dying because they lack ferritin, actually they drop on glucose very, very significantly, and those very few that actually survive, they will recover the glucose. So to make sense of these, we did what I think a posteriori is a fairly stupid experiment, which is let's compensate for the lack of an iron regulatory molecule by giving them glucose back. And that's exactly what we did. And Sebastian found out that he can actually restore the glucose levels in these animals. They now lack ferritin, so they should be dying. But actually, they all actually survived the infection. So this tells us that one of the major roles of ferritin by controlling iron metabolism is actually carrying a tremendous impact on glucose metabolism, which is important in the context of this infection. So we look again at pathogen load, comparing both aerobes and anaerobes, and we see absolutely no difference in any organ that we looked at. So we went back to the idea that this labile IM that we already shown was important for the pathogenesis of disease, could actually be the cause of deregulation of glucose metabolism. So what we did is to inject mice. We did a normal IM, which is IM with iron, or a protoporphyrin ring without iron, which is a synthetic molecule, but it just serves as a beautiful control for the role of iron in this model. And we're looking at blood glucose levels, and what we found is that IM by itself is sufficient actually to do a drop on glucose that is very similar to the drop of glucose that we see during sepsis, and this is completely dependent on the presence of iron. So somehow the iron that is coming inside the IM is important for this effect. We went back to the ferritin knockout mice and look at blood glucose levels once they actually receive the IM. And what we find actually is that the ferritin expression is important to control glucose levels once you're exposed to IM, suggesting that actually this iron must actually be compartmentalized inside the ferritin. What's perhaps more striking is actually that without ferritin, you can actually not survive. Without ferritin, you can actually not survive the injection of IM, really arguing again for the labile IM being important. So IM is a very interesting molecule for all living creatures. And one of the things actually that we have is the ability to sense perhaps as a surrogate of infection. So we classify it in this class of molecules that alert innate immunity for damage, and the name for that has been coined as alarmin. And this is actually really the work, it's not the work of our lab, it's the work of my very dear friend Marcelo Boz in Brazil, who identified some years back that heme can be actually sensed by TLR4, and this is not a contamination like in other set examples. There's really a specific sensing of heme. He also found that it can be sensed by NLRP3, and others have found actually that TLR4 in the endothelial cells can also do this. So based on the work of Marcelo, we decide actually to inject heme to TLR4 competent or deleted mice, and we look at glucose levels, and we see that this drop that we usually see when we inject heme into mice actually is completely TLR4 dependent, suggesting that perhaps even in vivo heme is being recognized by TLR4, but by no means the proof. Okay, so what is the molecular mechanism by which ferritin regulates blood glucose? We call that the million-dollar question. So we went back to the model of sickle ligation and puncture in mice that have or do not have ferritin, and we did target metabolomics in the liver to try to understand how this was going. And what we find actually is that if you have ferritin, you favor actually this whole cycle towards gluconeogenesis, that is the production of glucose in detriment of glycolysis. What I mean to say is that if you have ferritin, the hepatocytes after infection don't shift to glycolysis, and they keep producing actually glucose via the gluconeogenesis pathway. Then we did microRNA analysis, and ferritin is not a transcription regulator, so we were not expecting a lot of differences. And we had a small signal here for glucose 6-phosphatase C1, which is exactly the last step of the gluconeogenesis pathway. So this made sense to us, and we went back and tried to address the question whether ferritin would be actually regulating the expression of this protein, and more exactly on the expression of the complex, which is actually glucose phosphatase C1 in the ER lumen, but it requires glucose phosphatase transporter to bring actually glucose 6-phosphate into the ER and then put it out into the blood. So we did this initially in vitro. We did a luciferase assay using actually a reporter for glucose phosphatase C1. We expose hepatocytes to heme and TNF, and we look at luciferase activity, and we just reproduced data by other people showing that heme actually represses glucose 6-phosphatase C1 transcription, and this is actually enhanced by TNF, even though that is not shown in this graph. So we asked whether ferritin would be affecting this in the same system, and we see that just by overexpressing ferritin in hepatocytes, we increase glucose 6-phosphatase C1. This is a decrease when you have heme and TNF, and ferritin can actually hold better the expression of glucose 6-phosphatase C1 after this insult, maintaining actually at a level that is very similar to the basal one, suggesting that the expression of ferritin is actually, this is one of the mechanisms by which it enforces gluconeogenesis. So we did this also at the protein level with a series of adenovirus. The reason I'm showing you this is because when we express ferritin, we get more protein, but when we express the mutant ferritin, we don't get more protein. And this is a ferritin that cannot actually hold the iron and turn it into iron 3+. So the peroxidase activity of ferritin is very important. This is the normalization of that Western blot, and this is to tell that the two proteins are expressed at the same level. I'm realizing that I'm running out of time. So we went back in vivo and we looked what would be the effect of glucose, of sepsis on glucose phosphatase C1. We see that after CLP there's a tremendous decrease, and in the absence of ferritin there's actually 50% less glucose 6-phosphatase C1. We validated this by Western blot. We confirmed actually, we quantified all of this, and we see that there's a very significant decrease on the expression of glucose 6-phosphatase if we don't have ferritin, and and the activity of the enzyme is also reduced, really suggesting that there's less glucose being generated. I'm sorry, I did a mistake. This was the transporter, and now this is the glucose enzymatic activity. So I did a mistake here, probably. Sorry for this. So I'm just going to finish with this set of slides. So ferritin does control glucose 6-phosphatase, and we inquired whether liver gluconeogenesis is important to confer protection against sepsis. And the way we did this is to use a specific deletion of glucose 6-phosphatase in hepatocytes. We checked that the deletion was actually operational. And we now exposed the mice to a peritoneal contamination infection, which is similar to CLP, but we're now controlling the inoculum. And what we see actually is that mice that lack glucose 6-phosphatase C1 have a tremendous drop on glucose, and they develop this lethal form of hypoglycemia, showing that you require actually glucose coming out of the liver specifically to maintain blood glucose levels. And the survival actually of these mice is very, very poor once exposed to this type of events. And again, there's no changes in pathogen load, really arguing that what liver glucose production is doing is really maintaining homeostasis and not targeting directly the pathogen. So, as a conclusion, I think what we found here is that polymicrobial infection, I didn't mention this word before, is associated, and it's very well described, with a phenomenon called anorexia of infection, which is actually a defense mechanism that stops feeding behavior, and by doing that actually drops glucose levels in the circulation. By dropping glucose levels in the circulation, this has been shown, I think, very elegantly by the group of Ruslan and Metzentov to actually be a protective mechanism against microbial infection. And the idea is that you do an initial stage of hyperglycemia that I didn't show it, but we also have it. And then this hypoglycemia, or this lower glucose borderline hypoglycemia is actually essential for survival, and we can discuss the reasons for that. I think what our work adds to this is that if you have these type of events, which is red blood cell lysis in IM, you're now signaling perhaps via the TLR4 to do this, likely by the TLR4, producing reactive oxygen species in hepatocytes and blocking the activity of glucose 6-phosphatase and reducing further glucose levels, but to a point that it now becomes very, very deleterious. So the expression of ferritin here is absolutely required actually to bring back the glucose levels and restore homeostasis. And this is actually just written here, but I'm not going to repeat it for the sake of time. I would like to thank all members of the laboratory that contributed to this, and very particularly Sebastian Weiss that did all the initial work, and helped by Anne-Rita Karls for more the molecular biology part and granting sources for different grants. Thank you.